आइए अब हम बात करते हैं दूसरे मरहले की फॉर प्यूरिफिकेशन एंड आइसोलेशन ऑफ प्रोटीन्स। इस प्रोसेस के लिए हमारे पास दो ऑप्शंस हैं। या तो हम इस प्रोसेस को कंटिन्यू रख सकते हैं यूजिंग ट्रिटोन X100 या फिर हम इसको यूरिया के साथ परफॉर्म कर सकते हैं। इस पर्टिकुलर मॉड्यूल में हम बात करेंगे ट्रिटोन X100 की। हमारे पास सेल लाइजेट मौजूद है जो कि हमने प्रिपेयर किया था आफ्टर एंजाइमेटिक ट्रीटमेंट और एनी अदर ट्रीटमेंट। अब इस सेल लाइजेट में अगर देखा जाए तो सेल की हर किस्म की कंपोनेंट मौजूद है। दिस सेल लाइजेट कंटेन्स द साइटोप्लाज्म, cytoplasmic proteins, foreign proteins, DNA which has been digested, RNA, cytoplasmic organelles, and most importantly the broken pieces of the cell wall or cell membrane that caused the lysis. We want to separate the proteins from these unwanted materials. For that purpose we subject the cell lysate to centrifugation step. For 15 minutes we centrifuge it at maximum speed at 4 degrees centigrade in a microfuge. यहाँ पे एक चीज़ मैं बताता चलूँ कि प्रोटीन आइसोलेशन में टेम्परेचर प्लेज क्रिटिकल रोल। जितने भी स्टेप्स हैं प्रोटीन आइसोलेशन के उनको 4 डिग्री सेंटीग्रेड पे परफॉर्म किया जाता है। अदरवाइज देयर आर चांसेस दैट प्रोटीन ऑफ इंटरेस्ट may get denatured if we perform these steps at room temperature or at some higher temperature. Once the centrifugation is done, we separate the supernatant and we take the pellet and resuspend it in cell lysis buffer 2 at 4 degrees centigrade. And for that purpose we used 9 volumes of cell lysis buffer 2. After resuspension, we leave this cell lysate, resuspended cell lysate at room temperature for 5 minutes. And after 5 minutes, we are going to perform this centrifugation step once again. The 5 minutes incubation is basically the time which the cell lysis buffer 2 is going to perform its function, its action on the cell lysate and the components of the cell lysate. Later on, centrifugation at maximum speed for 15 minutes at 4 degrees centigrade is going to perform another separation. After this centrifugation, we are again going to separate the supernatant and we have the pellet. Now at this stage, this supernatant is equally important as is the pellet. We are going to test the supernatant as well as the pellet for our protein of interest. For that purpose I used here a word decant or transfer. I didn't said discard the supernatant जो कि normally हम करते हैं. So protein isolation में we are going to separate and transfer the supernatant into a separate tube, and we are going to resuspend the pellet in 100 microliter of water. Now we have two different samples. One is the resuspended pellet. The second one is the separated supernatant. We are going to test both these samples for the presence of our protein of interest. For that purpose we take 10 microliter from each samples in a new tube and add 2x SDS gel loading buffer, because we have to test the presence of proteins using gel electrophoresis. Once the loading buffer is added in these samples, now these samples are ready for gel electrophoresis, and we proceed further to run an SDS page and determine which sample carries our protein of interest. Up till this point we are successful to separate our foreign protein and to identify which of the separation, whether the resuspended pellet or the separated supernatant has our protein of interest. अगर यहाँ पे भी हमारे पास clear results नहीं आते, then we have an other choice कि इस process को थोड़ा सा extend कर लिया जाए and we can proceed further for solubilization of inclusion bodies. That is our step number 8 and some further steps. But if we are successful to identify at this stage which is our protein of interest, we can isolate that protein and we can proceed further towards purification.